Hello viewers. Today I am going to discuss on cells of immune system. I will discuss on the cells involved in immune system, their morphology and function. We know immune system is a defense mechanism of our body which protects us from foreign invaders. An immune system is composed of a number of specialized cells, organs, tissues, different protein receptors, and all these molecules play together to fight against the foreign molecule or foreign pathogen. Now, immune response can be innate or non-specific and specific or adaptive. Innate immune response, since it is a non-specific immune response, this immune response is same for all foreign pathogens. Whereas adaptive immune response has a memory, and once it is generated, it keeps the memory and it fights against the foreign pathogen next time very quickly. Now, depending on the components, immune response can be divided into cell-mediated immune response and humoral immune response. Now, all the blood cells of our body are generated in the bone marrow, and the process of generation of blood cells is called hematopoiesis, and it starts from a stem cell called hematopoietic stem cell, and approximately per day 10 to the power 11 to 12 number of new blood cells are generated. Now, hematopoietic stem cell can be differentiated into lymphoid progenitor cell or myeloid progenitor cell by two different pathways. Differentiation process depends on the amount and nature of the cytokines. Now, from the lymphoid progenitor cells, natural killer cells and T lymphocytes, B lymphocytes and dendritic cells, these are the cells are generated. From myeloid progenitor cells, the cells produced are monocytes, macrophages, all the granulocytes like neutrophils, eosinophils, basophils, also dendritic cells, they are produced. And all the white blood cells take part in immune response. All the granulocytes are also involved in immune response. The granulocytes are also called polymorphonuclear leukocytes because their nucleus is irregular shaped and divided into 2 to 5 lobes. Neutrophil, it has a 2 to 5 lobed nucleus and the cytoplasm is full of granules, and the granules can take both acidic dye as well as alkaline dye or basic dye. That is why neutrophil is so called. And the granules can be divided into two groups depending on the contents. Like the larger granules are called primary granules and they contain a number of enzymes like lysozyme, peroxidase, etc. Whereas smaller secondary granules contain lactoferrin, collagenase enzymes. And these enzymes and granules containing enzymes help in the digestion and destruction of the ingested pathogen, because neutrophils are also phagocytic cells and they are the first cells to reach a site of infection from blood. Eosinophil, another granulocyte, it has also granules in its cytoplasm, and these granules take the acidic dye eosin red. That is why they are so called eosinophil. And granules are full of different enzymes, RNAs, cationic peptides, and these enzymes are released mainly in case of a parasitic infection. And they also take part in allergic response. Eosinophils are also phagocytic cells. Another granulocyte is basophil. Basophil also has granules in its cytoplasm, and the granules take the basic dye methylene blue. That is why they are called basophils. And basophils also have granules full of different vasodilators like histamine, prostaglandin, serotonin, and these molecules act as vasodilators. That means they increase the diameter of the blood vessel, and they also act in case of an allergy. Now, macrophages are 3 to 5 times larger than monocytes, and they are generated from monocytes, and they have more organelles in their cytoplasm compared to monocytes, and macrophages are present throughout the connective tissue and in the membrane surrounding the small blood vessels. And macrophages can be tissue resident or they may be free in the interstitial fluid. And depending on the tissue where a macrophage is present, it is named differently. Like when a macrophage is present in liver, it is called a Kupffer cell. When a macrophage is present in spleen, it is called sinusoidal cell. And macrophages play a very important role in removing the foreign pathogens. How? By phagocytosis. These cells can do phagocytosis. Okay, and on the surface of macrophages, there are different types of receptors. Receptors for mannose sugar or for Foucault's, or a special kind of receptor called toll-like receptors. All these receptors are present on macrophage surfaces. They can bind to specific ligands present on the microorganism cell surface. And those ligands are called pathogen-associated molecular pattern. When these receptors of macrophages bind to the specific ligand, it triggers phagocytosis. That means the foreign pathogen is engulfed and macrophage chew it. Now another phagocytic cell of immune system is called dendritic cell. Dendritic cells are present in all those tissues which are in direct contact to the environment. That means in the lining of nose, nasal cavity, in the GI tract, in the respiratory tract, in the genitourinary tract. Okay, and they act as sentinels of immune system. And whenever an antigen enters into our system, dendritic cells capture them. Then dendritic cells migrate to the nearest lymphoid tissue like lymph node or spleen, and they present the antigen to other cells of immune system, mainly to T lymphocytes. Now T lymphocytes get activated and they differentiate. Another very important cell of immune system is mast cell. Mast cells are generated from their precursors. These precursor cells, they come to blood. In blood, they mature into mast cells. Mast cells then move to different tissues. Mast cells also have granules in their cytoplasm, and the granules are full of different vasodilating molecules like histamine, prostaglandin. So mast cell also takes part in allergic responses. They are non-phagocytic cells. Now I will tell you about lymphocytes, very important cells of immune system. They constitute almost 20 to 40% of white blood cells. And lymphocytes can be divided into three groups like T lymphocytes, B lymphocytes and natural killer cells. T lymphocytes are generated in bone marrow like all other blood cells. Then some of the lymphocytes move to or migrate to a gland called thymus, which is present above the heart. Now in the thymus, those lymphocytes mature. That is why they are called T lymphocytes. So T lymphocytes actually generate in the bone marrow and mature in the thymus. From thymus, they come to the blood. And during their maturation in thymus, they express receptors on their surface. These receptors are called T cell receptors. These receptors play an important role in the immune response. T lymphocytes can be of many types like T helper cells, T C cells or T cytotoxic cells, T regulatory or suppressor cells. T helper cells can recognize antigen presented by a specific surface marker protein MHC class II on the surface of phagocytic cells like dendritic cells, macrophages. When a T helper cell recognizes that complex, it gets activated and it starts differentiation. Now after differentiation, they produce a clone of cells. Some of them are called effector T lymphocytes, some are called memory T lymphocytes. Effector T lymphocytes show the effect immediately. They secrete a number of molecules called cytokines. These cytokines activate the other immune cells, and memory cells are kept for future. Now I will tell you about the TC cells, T cytotoxic cells. These cells also have receptors on their surface, T cell receptors, and these receptors can recognize self-altered proteins or viral proteins presented on almost all nucleated cells by a surface marker protein called major histocompatibility complex 1. And if a receptor present on a tissue cell recognizes this complex, that tissue cell gets activated, differentiates into effector cells and memory cells as Th cells. Another group of T lymphocytes is called T regulatory cells. They actually monitor the presence of self-reactive T cells in blood. I told you that all the self-reactive T cells are deleted in the thymus to ensure that only self-non-reactive T cells come to blood. But if by mistake some self-reactive T cells come to blood, then they are monitored by these T regulatory cells and they check the growth of those self-reactive T cells. Otherwise, self-reactive T cells may lead to autoimmunity, where our own immune system acts against our own components. Now I will tell you about B lymphocytes. B lymphocytes are generated in bone marrow, but the alphabet B came from bursa of Fabricius. It is an organ present in birds, which is the prime site for B lymphocyte generation in birds. Later it was found that in mammals, bone marrow is the site for B lymphocyte generation. Now mature B lymphocytes, they display on their surface the receptors called B cell receptors, which are also called membrane bound immunoglobulins. The effector B lymphocytes are converted to plasma cells. Plasma cells secrete soluble immunoglobulin molecules. Now another cell which is generated from the lymphoid progenitor cell is called natural killer cell. Natural killer cell is a part of innate immune system. Natural killer cell directly kill the tumor cells or virus infected cells. Now so far the cells we have studied, 3 of them are called antigen presenting cells: B lymphocytes, dendritic cells and macrophages. These 3 cells are called professional antigen presenting cells of immune system. Now organs of immune system can be divided into 2 groups: primary organs and secondary organs. There are 2 primary organs present in our system: bone marrow and thymus. And primary organs are the site where the lymphocytes generate. And secondary organs include spleen, lymph node, also mucosa associated lymphoid tissue. Spleen and lymph node, these are 2 secondary lymphoid organs which are well organized, whereas mucosa associated lymphoid tissue is less organized secondary lymphoid organ. And secondary lymphoid organs provide sites where lymphocytes meet the specific antigen and then mount an immune response. Between the 2 primary lymphoid organs, bone marrow is the site for hematopoiesis. That means it is the site for generation of all blood cells, and at the same time it is the site for B lymphocyte generation and maturation. If we look inside the bone marrow, then we will see that many different blood cells are present in the bone marrow, and there are some fat cells, also epithelial cells and other stromal cells present. And these stromal cells, they provide a 3D network where the immature blood cells get shelter and many factors secreted from the stromal cells help in their maturation. Second primary lymphoid organ is thymus. Thymus is located above the heart. It is a bilobed organ, and each lobe is surrounded by a membrane called capsule. Now projections from the capsule enter into the thymus and divide the thymus into several lobules or small compartments. Each lobule is divided into two parts. The outer part is called cortex and the inner part is called medulla. The cortex is full of immature T lymphocytes or called thymocytes, and the medulla part is full of different cells like macrophages, dendritic cells, epithelial cells, T lymphocytes, etc. Now the composition of thymus, weight and composition of thymus decreases with age. The weight of thymus is maximum at puberty, and then both the cortex and medulla part decreases with time. Now I will tell you about the secondary lymphoid organs. Two well-organized secondary lymphoid organs are lymph nodes and spleen, and less organized organ is mucosa-associated lymphoid tissue. Now spleen is located in our abdomen, left abdomen, and it is surrounded by a membrane called capsule. From capsule, long projections enter into the spleen and it divides the spleen into many compartments. And if we look into each compartment, we will see two zones, one red zone, one white zone. The red zone is called red pulp and the white zone is called white pulp. The outer region or the red region is full of macrophages. These macrophages capture the old and damaged RBC, and that is why, due to the presence of porphyrin ring and iron deposition, this region looks red. And the white region is full of T lymphocytes and B lymphocytes. B lymphocytes are arranged in concentric circles, and these circles are called primary follicles. Spleen has no lymphatic vessel inside it. It has only splenic artery and vein. So all the blood-borne antigens are trapped in the spleen. Okay. So all blood-borne antigens or antigens that enter into our blood system, blood circulation, get trapped in the spleen. Now lymph nodes are present at the joints of the lymphatic vessel. Where lymphatic capillaries meet, a lymph node is present, and almost 500 to 600 lymph nodes are present throughout our body, and they are mainly found at different joints, different joints and as clustered, and their function is to trap antigens coming through lymph. So spleen traps all blood-borne antigens and lymph nodes trap all the lymph-borne antigens. Now I will tell you about mucosa-associated lymphoid tissue. It is a less organized secondary lymphoid organ and it is present under the mucous membrane. Now the mucous membrane present in our GI tract, respiratory tract, urogenital tract has a surface area of almost 400 square meter, and these are the portals of entry for most of the pathogens. Okay, and these portals of entry are defended by tissue called mucosa-associated lymphoid tissue, and this tissue is composed of well-organized clusters of lymphoid cells, mainly T lymphocytes and B lymphocytes. And one good example of mucosa-associated lymphoid tissue is Peyer's patches. In our ileum, we can see patches of primary follicles arranged in organized manner. Now, what are primary follicles? Primary follicles are made of concentric circles of B lymphocytes. So, B lymphocytes arranged in concentric circles are found in the Peyer's patches. It is called Peyer's patches because it was discovered by scientist John Conrad Peyer. Also, in our GI tract, the mucus-associated lymphoid tissue is present just below the mucosal epithelium layer, and the MALT or mucosa-associated lymphoid tissue in gut is called gut-associated lymphoid tissue or GALT. Similarly, the mucosa-associated lymphoid tissue of respiratory tract is called BALT, that is bronchial-associated lymphoid tissue. Now, in the mucous membrane, in the mucosal epithelium, we can see some cells called M cells or microfold cells. Microfold cells have a large pocket in their basal surface. The pocket is full of different lymphoid cells like B cells, dendritic cells, macrophages, B lymphocytes, and the apical surface has tiny invaginations called microfolds, and these microfolds, they can engulf an antigen. So after engulfment or phagocytosis, the antigen comes into the pocket full of different cells. The antigen is phagocytosed by the antigen presenting cells, presented to the T cells. So very quickly the immune system can respond against that antigen. Tonsil and adenoid, they are also examples of mucosa associated lymphoid tissue. Now I will tell you about the receptors involved in immune system or immune response. There are some preformed receptors present in the immune system, and some receptors are somatically modified. And binding of a ligand to its specific receptor initiates an event for immune response. Now among the preformed receptors, pattern recognition receptors are very important. Also there are receptors for Fc region of immunoglobulins, and they are called Fc receptors. Also KIR and CAR receptors present on natural killer cell, complement receptors, cytokine receptor. There are many preformed receptors, and mainly they are part of innate immune system. Whereas B cell receptors and T cell receptors, they are somatically modified receptors, and they are part of the adaptive immune response. B cell receptors are membrane bound immunoglobulins, and each immunoglobulin molecule is composed of 2 heavy chains and 2 light chains. Now if we look at the cytoplasmic tail of the membrane bound immunoglobulin, then see it is too short to transduce any signal. So the membrane bound immunoglobulin needs help from 2 other proteins called Ig alpha and Ig beta. So as soon as a B cell receptor or membrane bound immunoglobulin binds to an antigen, some conformational changes occur at the membrane bound region, and it activates the Ig alpha, Ig beta, and it transduces the signal. Now as soon as a B cell gets activated, it differentiates into plasma cells and memory cells. Plasma cells secrete immunoglobulin molecules of same specificity, whereas T cell receptors they are also membrane bound receptors, and they are made of 3 pairs of peptides: alpha, beta, gamma, delta, and 2 epsilon. T cell receptor recognizes antigen presented by MHC molecules. MHC molecules are major histocompatibility complex molecules. All nucleated cells present on their surface MHC class 1 molecule, and APC, antigen presenting cells present on their surface MHC class 2 molecule. And these T cell receptors present on a Th cell or T helper cell recognizes antigen presented by class 2 MHC molecule, whereas T cell receptor present on a TC cell or T cytotoxic cell recognizes antigen presented by MHC class 1 molecule. So B cell receptors directly bind to the antigen, whereas T cell receptors indirectly bind to antigen. That means when the antigen is presented by MHC molecules, only then they can bind to it and recognize it. And in case of T cell receptor, the signal is transduced by the accessory molecules, and the accessory molecules together is called CD3 molecule.